All right. Welcome, Jeff Woodhead. Welcome back to all of our participants who probably just came out of a wonderful complement sim overview from Liesel Shota. Quick introduction here. Thank you again for joining us and staying with us in the discovery and preclinical track at MIDD Plus 2023. In this session, Jeff Woodhead, one of our senior scientists from our DILIsym Services division, will present an overview of a recent QST poster that he offered. As a reminder, again, ask questions in the Q&A tab. If you have technical issues, throw those in the chat feature. There's a poll question. We'd love your participation. That should auto-launch. Without further ado, Jeff Woodhead, everyone. Thank you.
Thanks, Andrew. So today I would like to be talking about some of our work on the DILIsym modeling of Verdiperstat, which is the first-in-class myeloperoxidase inhibitor that we ran through DILIsym for a client a couple years ago. So Verdiperstat is a drug that's been used or in trials at least for treatment of multiple system atrophy or MSA, and for Lou Gehrig's disease ALS. And at phase two, this has shown some efficacy at this 600 milligram BID dose, but that's a pretty big dose, right? That's 1.2 grams a day is a dose you often don't get to, especially if you're in the liver tox field. You're not really wanting to get to that point. And there's some in vitro signals they were a little bit concerned about, and a couple of clinical signals where they kind of had to raise an eyebrow at them. So they wanted to run it through DILIsym before moving on to phase three, just to make sure, well, we want to be extra careful making sure this isn't going to kill anybody, right? So they had some this is a company that we've worked with in the past. We've had a lot of prior success with them. There was a recent paper that just came out on the CGRP compounds, the Japants, I guess, is what their suffix is. And Biohaven worked with us on some work for those compounds. And there's also, I think, the Riluzole, which has a paper that's out as well. So we'd worked with this company before, and they decided to come to us and hey, could you guys look at this compound for us? And so essentially we did. The basic idea here is that we ran it through standard DILIsym workup with all of our in vitro experiments, did a PBPK model for it, and put it through DILIsym and found that it's deeply unlikely that it was going to cause hepatotoxicity, at least from the um, for the mechanisms that DILIsym includes, right? Uh, so going through the actual work that we did, we in DILIsym, for those of you who aren't familiar with it, we represent three main mechanisms of toxicity, looking at generation of reactive oxygen species, initiation of mitochondrial dysfunction and toxicity, and disruption of the process for transporting intracellular bile acids, and then thus causing intracellular bile acid buildup. So we looked at our in vitro assays for both of those and, well, ended up with some signals, at least in each of those mechanisms. So, well, it's a lot of parameterization we're going to have to do. Fortunately, the bile acid transporter studies, not a ton going on there. That is the MRP4 was really the only signal we had there. So we parameterized it, but you can look at this and say, well, probably not going to cause a whole lot of accumulation there because MRP4 isn't used a ton during the normal homeostatic conditions. So
Jeff,
looking,
sorry,
yes,
sorry to interrupt. Could you put your slides into presentation mode for us?
okay, is it not in presentation mode?
It's not in presentation mode, and we're still on the first slide here.
That's a little bit weird. I'm on slide 7. um, what do you see now?
It looks like we are on slide 7, but it's not in presentation mode.
Okay, now.
Still not in presentation mode. Still on slide 7.
Okay, well, all right. I'm gonna try to share again. Share screen. Let's see if I can just share the entire screen. How about that? Uh, Here you are. How about that?
Much better. Thank you so much, Jeff.
Okay, so um, I'll just go back here since it was kind of the first results that we've really seen. Uh, this was the in vitro results for bile salt transporter inhibition. You can see some inhibition on MRP4, but nothing on the other three bile salt transporters. So you put an IC50 in there, about 32 micromolar, fairly high IC50, but again, a fairly high dose drugs. So um, you want to put it in there just to see what it's going to do, right? Um, you also saw some mild inhibition of the electron transport chain with in vitro. What we're looking at here is the Seahorse XF analyzer results from uh, Verdiperstat uh, compared to the measured intracellular concentration. These are intracellular concentrations that are measured by mass spec in the cell lysate versus the oxygen consumption rate. And you can see a pretty consistent oxygen consumption rate as percent concentration goes up. It's not a huge one, but it's there. And so we ended up parameterizing that as well. The red line here is what we simulated as the drop in OCR, and thus, as a result, the parameters that we used for ETC inhibition. The second was oxidative stress, and again, you can see a minor signal here, but it's there, right? You're seeing a little bit of an increase in oxidative stress towards the higher concentrations here in the in vitro assay. So we put a parameterization on that as well, and that was the parameter value we put down here. So we have parameter values now for each of our three mechanisms: transporter inhibition, oxidative stress, and mitochondrial dysfunction. And from there, we're able to put together a simulation. But on the way, there's every simulation project we say here there's a curveball, and this was our curveball. When we looked at the mass spec data, we saw some really significant accumulation in the cells. That the concentration of verdiperstat within the cells that we measured was much higher than the nominal concentration in each particular um, assay. That didn't jibe with what the PBPK model and the physicochemical properties seemed to suggest, which was a KP of liver that was a lot closer to 1. So we went back and we reparameterized the mitochondrial uh, assays and the ROS toxicity using a intracellular concentration of assuming that the KP was 1, right? So that the partitioning between the in vitro media and the cells was at the ratio of 1. So using that new uh, intracellular concentration estimate, we reparameterized everything. Uh, and what that does is that because now there's less drug in the cells, that means that the toxicity mechanisms are a little bit more sensitive, right? Because you're having the same amount of activity at lower intracellular concentrations. So now you've kind of upped the sensitivity of the assay a little ways, the decrease the predicted concentration at which these effects are going to start kicking in. So this is kind of a worst case scenario, right? That if we assume that something went wrong with the LCMS assay, and if we assume this is just kind of covering our bases, right? Like there's the potential that it is this sensitive. So let's parameterize it here. Let's take a look at it and see if there's any difference in the predictions with this more sensitive set and the original parameterization. So now we have these two uh, parameterizations here, the primary value, which we just went through, and then the alternate value, which was this more sensitive parameterizations. You can see that we're about 100-fold more sensitive for both oxidative stress and mitochondrial dysfunction. So running through the primary parameterization, going all the way up to 600 milligrams BID, we saw nothing. There was what they did see in the clinic some mild liver enzyme elevations at these lower doses in phase 1 for 10 days, but it wasn't entirely clear whether they were they were liver related or actually drug related or not, right? They saw the elevations there, then they went through phase 2 and saw no ALT. So it kind of confused them a little bit. We were trying to see whether those were likely to be drug related or not. And we saw unlikely that there's anything there for either of those protocols, and certainly even that for the 600 milligram BID that they're targeting for phase 3. Um, and so you can look at the EDISH plots here. These are plots of fold change in ALT uh, over the upper limit of normal on the bottom, and fold change of bilirubin over the upper limit of normal on the y-axis. Uh, the idea of an EDISH plot being that if you see something going into this bottom right quadrant, then there's some potential danger of liver injury. And you see things going into this top right quadrant, that means there's significant elevations in both ALT and bilirubin, and you're really likely to have some liver injury issues, and you should not go there. But as you can see, the simulated EDISH plots that we've produced here, nothing has moved even. Everything is just stacked on top of each other in that one little dot where there hasn't been any shift in ALT or bilirubin. So what about the alternate parameterization? Well, what we did see is, you start seeing a little bit of movement at these higher doses. With this more sensitive parameterization, you start seeing one individual drift away from the pack here and get some very mild ALT increases and very mild bilirubin increase as well. Uh, so what that suggests, even at these really high doses and at this parameterization that we feel like is at the absolute higher bound of the potential sensitivity to liver injury here, you're not seeing anything clinically significant. You're seeing this very mild ALT elevation that's really going to come out in the wash. It's not going to it's not really significant liver injury. You're not losing very many of your cells there. Individual with a one-fold ULN increase in their ALT elevations probably wouldn't notice it unless they are doing their blood chemistry, right? So, um, nothing really is going to be particularly significant, at least from our predictions and based on the mechanisms that we are able to represent. Um, So they went forward with the phase 3 clinical trial, and good news, no significant liver signals were observed, which is what we predicted. This was of course, a prospective prediction of safety, and then we turned around and we saw safety in the clinic. Unfortunately, they also didn't see efficacy, so they ended up terminating the program for MSA. They're still looking at it in ALS though, so we don't have the results of that phase 3 study yet. Um, so hopefully for them that comes out well. But uh, from a DILIsym perspective, this is what we wanted to see, right? We predicted safety, the drug was saved. So that was the end of the story here, and story of how DILIsym was used to predict prospective safety in a uh, developmental compound, and how those predictions were borne out by future clinical trials.